My name is Brianna Sciuto. I'm a senior at Carmel High School on the Monterey Peninsula, and this summer, with the guidance of Dr. Kelly Moulaine, I conducted research on Monterey Bay giant kelp forests and their potential to mitigate the impacts of climate change on local marine ecosystems. I wanted to do this project because I'm passionate about environmental health and regeneration, and I especially want to preserve the incredible ecosystems in my home in Monterey. This project allowed me to use my skills to contribute to and summarize research regarding oceanic health and potential solutions to climate change related issues. Today, I'm just going to provide some context and then present the methods and results of my research. Now, I'm sure you've all heard that the ocean makes up 70% of the Earth's surface, but it's kind of hard to comprehend what that actually means. It means it produces about 50% of the oxygen we need. It means it takes in 25% of the greenhouse gases that we emit, and it captures 90% of the heat that those emissions produce. We tend to see the ocean as separate from our lives, but our lives depend on it. However, it sustains its own damages when it protects us from climate change. Now, these three problems tend to present themselves. First, high ocean acidification occurs when we emit excessive amounts of carbon into the atmosphere, and it dissolves into the ocean, forming carbonic acid, which is comprised of CO2. As a result of this, the ocean becomes, on average, more acidic, which, as you can imagine, hurts entire ecosystems. Ocean deoxygenation, which is the decrease in average dissolved oxygen in the ocean, occurs as a byproduct of global warming. As water temperature increases, its ability to hold oxygen decreases, so the plants and animals in the ocean are left with less oxygen. Finally, eutrophication occurs when there are excess nutrients in the water, which is actually a bad thing. They lead to harmful algal blooms, causing dangerously low oxygen levels in certain regions. Now, the ocean relies on a precise homeostasis to sustain the complex biogeochemical processes, interactions, and systems that occur under the surface, so even a slight change in the chemistry of the water is significant. So we have all these problems in our ecosystem. My goal with this research was to determine if kelp forests could be a potential solution to these problems. Specifically, I wanted to know, to what extent does the presence of kelp forests mitigate the impacts of anthropogenic ecological threats in the region? To do this, I swam out and I took samples in the water in 2 kelp forests in Monterey Bay. I collected water from the center of the kelp forest, at the edge, and outside of the forest at different depths. And I compared the chemistry of each of the samples to see how the kelp influenced the water. I tested dissolved oxygen, pH, nitrates, and phosphates. This is because pH can tell us whether kelp forests mitigate ocean acidification. Dissolved oxygen, or DO, indicates whether they mitigate deoxygenation, and nutrient levels indicate whether they mitigate eutrophication. These are the sample sites at each kelp forest. Now, before diving into the results, I want to talk about kelp. Kelp forests are among the world's most productive ecosystems, providing coastal protection and biodiversity. And what researchers have been finding is that they could serve as an important tool in the fight against climate change. What's most important to know is that kelp helps absorb CO2 and emits O2 as a product during photosynthesis. Because of their high productivity, this process occurs rapidly and on a large scale, making it more fit to combat climate change within the ocean. Because kelp absorbs CO2, which we established earlier is a primary component of carbonic acid, the ocean should be less acidic near kelp. We also expect to find higher dissolved oxygen and lower nutrient levels due to kelp's absorption capabilities. These are the 2 kelp forests where I collected the data. My methods were pretty standard. Using scuba gear, I collected water samples from the kelp forest center, edge, and 10 meters outside at 3 depths, the surface, 4.6 meters below the surface, and 9.2 meters below the surface. Each test was conducted in triplicate, so we analyzed the mean values, and we analyzed all data points in 2 key ways. We compared the data between the center, edge, and outside of the kelp forest to analyze the kelp's impact in relation to the kelp forest itself. We also compared the data between depths at each site to analyze how the kelp's impact changed with depth. I left out the more complex statistical analysis for the sake of this presentation. Now, first, dissolved oxygen. Dissolved oxygen exhibited substantial variation, but the values only varied in a significant and consistent manner between sites at the surface. This is to be expected because the majority of photosynthesis occurs at the surface in kelp forests, and ultimately, DO was consistently higher at the center of the kelp forest as compared to the edge and outside. Surface samples consistently exhibited higher DO than samples at depth, and that variation was considerably more pronounced at the center of the kelp forest. Basically, as you can see with these graphs here, the difference between the surface and bottom samples was greater within the kelp forest. We also consistently observed a pronounced increase in pH at the center as compared to outside of the kelp forest. pH was consistently higher at the center of the kelp forest as compared to the edge and outside. Similarly to the DO values, there was a noticeable trend in the difference between the surface samples and the samples at 9.2 meters below surface level. They were substantially and consistently higher in the center than at the edge and outside. Nutrient levels in Monterey Bay are generally low, even during upwelling season, and the nitrate and phosphate test strips that we have had a detection limit, so that limited our ability to accurately test the nutrients. But we detected phosphates outside of the kelp forest at depth, and nutrients never appeared at the center of the kelp forest at the surface. Throughout the study, no nitrates were detected, and there's actually a scientific reason for this. Lower nitrate levels should be expected in Monterey Bay because it has internal bores, which are just subsurface waves. The bores are unique to Monterey and they contribute to high energy mixing. You may have heard the saying, the solution to pollution is dilution, and that's exactly what happens with nutrients here. The high energy mixing immediately dilutes the nutrients being introduced into the system, which prevents any significant changes to ecosystemic health, such as those produced by eutrophication. This is opposed to ecosystems such as that of San Francisco Bay, which is depicted at the top here, in which nutrients are allowed by the stillness of the water to concentrate in a single area, creating eutrophication. Now, that's a lot of information to make sense of. Analyzing the DO and pH data between the center and outside can indicate whether or not the kelp forest's rate of oxygen output and carbon intake is substantially affecting the chemistry of the surrounding water. Basically, this answers the question, does the photosynthesis of the kelp actually have an impact on the water? The DO and pH levels outside of the forest provide a control group that gives us an idea of the chemistry of the unaffected water, and the levels inside of the kelp forest give us an idea of the extent to which the kelp's photosynthesis influences that chemistry. Now, I analyzed the pH and DO values together in this presentation because they were highly correlated, which indicates that the kelp's photosynthesis is what caused these differences. Higher pH and DO levels indicate that kelp forests significantly impact surrounding water chemistry, helping to mitigate ocean deoxygenation and acidification. And analyzing the data between depths is also valuable because it confirms the impact of the kelp canopy. The vast majority of the productivity of kelp forests occurs in the upper 3 meters of the water column. So measuring the difference in DO and pH levels between depths is an alternative means of determining the mitigating impacts and potential of kelp because, like comparing it across locations, it allows us to compare these measurements in regions where the water column where kelp biomass is different. The substantially higher pH and DO near the surface indicate the high primary productivity and mitigating potential of the kelp's canopy. And that's a seal friend that I made on one of my 3 dives in sampling. So overall, I found that kelp is an indispensable management tool in ecosystems in which it resides because it locally reduces the impact of climate change. As such, there should be efforts in local communities to fund the conservation of our giant kelp forests, so that they can continue to provide ecosystem services that protect our bays, our wildlife, and ourselves. Thank you.
Great job, thank you. So what are some next steps you'd like to take with your research?
I will be publishing my research paper this month, and I also kind of want to use it to educate people in Monterey because it wasn't until I was educated about kelp that I realized how important it was, and I realized how passionate I was about oceanic health. So I think using this at the middle school in Carmel and at Carmel River School, which is our elementary school, can help introduce the idea of kelp as it impacts our local ecosystems.
That would be really great. Another question, did you see a significant difference between the two kelp forests?
I did actually. So I noticed that Maccabee kelp forest was a lot denser than breakwater kelp forest. These were my two kelp forests. And breakwater was nearer to a source of pollution, a pipeline. So breakwater was higher in phosphates actually, and Maccabee kelp forest exhibited a lot higher mitigation potential. So the kelp signal extended uh, beyond the kelp forest itself and into the edge samples, as opposed to breakwater where all of the mitigating potentials seem to be concentrated around the kelp forest.